Good evening, my name is Mihib Greywall. Over the summer, I spent 8 weeks studying liver cancer using the zebrafish model in the Sadler lab at Mount Sinai. Liver cancer is the third leading cause of cancer-related mortality worldwide, but unfortunately there is no cure for it. Last year, I did a project on organ donation rates, which remain extremely low in our country. My project made me realize how important it is for us to develop alternative treatments for liver cancer who are not as dependent on organ donors for liver transplants. The gene I focused on in my experiments was UHRF1. UHRF1 is a protein-coding gene and an epigenetic regulator that is involved in DNA methylation and histone modification. It is also required for liver growth and regeneration. However, UHRF1 is highly expressed in patients with advanced liver cancer. Previous researchers in my lab have confirmed that UHRF1 is in fact a driver of liver cancer, but the exact mechanism of its oncogene function is still very unclear. So, in my study, I analyzed transgenic zebrafish livers to elucidate how overexpression of UHRF1 correlates with hepatocyte cell division and contributes to the development of liver cancer. So, just to start off with a few basic definitions, epigenetics is a study of heritable changes in gene expression that are not caused by actual alterations to the DNA sequence. One important epigenetic system is DNA methylation. DNA methylation is the addition of a methyl group onto the cytosine base. During DNA replication, UHRF1 recognizes hemimethylated DNA and recruits an enzyme called DNA methyltransferase to maintain methylation in daughter cells. A transgenic organism has a gene from another organism artificially inserted into its genome. Zebrafish happen to be ideal model organisms because not only are they vertebrates, but they're estimated to be 60 to 75% genetically homologous to humans. Further, they have very large progeny. Some of the fish I was working with were laying 200 to 300 eggs at a time, and their embryos are transparent, which makes them very easy to assess and genetically manipulate. I worked with transgenic zebrafish that overexpress human UHRF1. The human UHRF1 is tagged to a green fluorescent protein abbreviated as GFP, so that it's easy to locate and analyze. It was under the hepatocyte-specific promoter FABP10. So for the purpose of my study, I refer to these transgenic zebrafish that overexpress human UHRF1 as UHRF1 high, and in many of my analyses, I compare them to wild-type zebrafish. So my first hypothesis was that in UHRF1 high zebrafish embryos, the level of UHRF1 would increase between 3 and 10 days past fertilization due to increased activity from the FABP10 promoter. Along with this increase in expression, I hypothesized that nuclear morphology and cell division be affected in the UHRF1 high livers. So while previous researchers have begun to look at UHRF1 expression at older time points in adult zebrafish, in my study I really focused on the embryonic development to see how the level of UHRF1 is changing in the days that possibly precede tumorigenesis. So first, in order to analyze how UHRF1 overexpression affects cell division, I treated 3, 4, and 5 days past fertilization, both wild-type and UHRF1 high zebrafish embryos with BRDU. BRDU is a synthetic nucleoside used to tag cells that are dividing. I then anesthetized them and fixed them. My mentor provided 10-day-olds, both wild-type and UHRF1 high zebrafish embryos that were previously incorporated for the BRDU. I then conducted immunofluorescence on all the fixed embryos in order to detect the BRDU nucleoside. I used a secondary antibody to amplify the signal of my primary antibody so that I could easily see which cells were positive for BRDU. I then dissected out the livers out of the fixed embryos. I stained them all with HOX, which is a DNA stain. I then mounted them onto slides and imaged all my liver images using all my liver samples using a Leica confocal microscope, which really felt like a spaceship to me. I then analyzed all my liver images using a program called ImageJ. For each time point, I measured approximately 10 wild-type livers and 10 UHRF1 high livers, and within each liver, I set the program to analyze 100 to 200 hepatocyte nuclei. The conditions I measured for were UHRF1 expression via the GFP fluorescence, cell division via the BRDU fluorescence, DNA staining intensity via the HOX fluorescence, the nuclear area and circularity of the nuclei. So in this first panel here, I have the wild-type livers on the left and the UHRF1 high livers on the right. What you are seeing are the hepatocyte nuclei in the DNA stain channel. All of these are from 3, 4, 5, and 10 days past fertilization. So as you can see here in this first box in this plot, I compare the nuclear area in micrometers. The green boxes represent the UHRF1 high livers and the black represent the wild-type livers. Interestingly enough, the nuclear area was significantly higher in the UHRF1 high livers at all 4 time points. I hypothesized that perhaps the nuclear area was larger in the UHRF1 high livers because they had a greater DNA content. I did some further reading on the Hox DNA stain, and I learned that it actually intercalates into the DNA because it binds to the minor groove of the DNA strand. So, since I had the average Hox fluorescence and the nuclear area, I realized that I could multiply these two values to approximate the total DNA content within hepatocyte nuclei. So I would then be able to compare the relative total DNA content between the wild-type and UHRF1 high livers. As you can see here in the second box and whisker plot, this relative DNA content was in fact higher in the UHRF1 high livers at 3, 5, and 10 days past fertilization, which bolstered my hypothesis. I also read that Hox fluorescence is usually bright when chromatin is tightly packed and dim when chromatin is loosely packed. As you can see here, the Hox fluorescence was slightly dimmer in the UHRF1 high livers at all 4 time points, which is suggestive of a looser chromatin structure, or euchromatin, which may be somewhat counterintuitive considering the fact that I personally felt that an overexpression of UHRF1 would lead to tighter chromatin structure, or heterochromatin, and this summer I will definitely be looking further into this. In this last figure here, I compared the circularity of the nuclei. I measured the circularity on a scale from 0 to 1, with 1 being a perfect circle. As you can see here, the hepatocyte nuclei in the UHRF1 high livers were significantly less circular, particularly at 10 days past fertilization. In this image here, you can see that the nuclei are extremely abnormally shaped and distorted, and they barely resemble their wild-type cohort. Since abnormal morphology is always a hallmark of cancer, I was starting to believe at this point that tumorigenesis begins to set in at 10 days past fertilization. In the second panel here, I have the wild-type livers on the top and the UHRF1 high livers on the bottom. The blue represents the Hox DNA stain, the red represents the BRD fluorescence for cell division, and in the UHRF1 high livers, the green is representative of the green fluorescent protein that the gene is tagged to. I compared the mean percentage of dividing hepatocytes between the wild-type and the UHRF1 high livers. As you can see here, there is no significant difference in cell division at 3 days past fertilization, perhaps because the gene has just been turned on at this time point. However, at 4 days, while there was still 17% cell division in the wild-type livers, there was only 1.3% in the UHRF1 high livers. Further, by 5 days past fertilization, while there was still 12% cell division in the wild-type livers, there was close to no cell division in the UHRF1 high livers. However, at 10 days past fertilization, while cell division had stopped in the wild-type livers, it was certainly beginning to pick up again in the UHRF1 high livers. Seeing these anomalies in cell division led me to try to investigate further in simply the UHRF1 high livers. In this last panel here, I have 4 UHRF1 high livers at the 4 measured time points. Here I have the livers in solely the green fluorescent proteins channel. So I compared the level of UHRF1 expression, and I found that it gradually increases up to 5 days. 5 days is when the UHRF1 high liver is at its max, shown by how much brighter these nuclei are compared to the other 3 time points. Then the level of UHRF1 begins to decrease, shown by the fact that these nuclei are clearly dimmer than these nuclei. I then analyzed an individual 4-day UHRF1 high liver and an individual 10-day UHRF1 high liver. I divided the hepatocytes based on whether they were positive or negative for cell division. Interestingly enough, I found that those cells that were positive for cell division had a very low level of UHRF1, which led me to believe that the level of UHRF1 is inversely correlated with whether or not a cell divides. I summarized my findings in terms of cell division and UHRF1 expression into these 2 graphs. As you can see here, the level of UHRF1 gradually increases up to 5 days and then decreases, while the level of cell division does the exact opposite, with the lowest point of cell division being at 5 days and then beginning to pick up again at 10 days. So my current working model is that UHRF1 overexpression leads to senescence at 5 days. Senescence is when a cell loses the ability to divide. This senescence may be related to the hypomethylation that I was seeing in this first panel. Then I believe that tumorigenesis begins to set in at 10 days past fertilization, due to the fact that I saw an increase in cell division and very abnormal morphology. This summer I will be returning to the lab to test additional time points between 5 and 10 days past fertilization to see whether cells are either escaping tumorigenesis or whether some cells are not even senescing and straight becoming cancer cells. Additionally, I will be testing time points after 10 days past fertilization to see if and how the tumors actually develop. Further, I will be learning how to conduct flow cytometry so I can more accurately measure the DNA content and gauge which phase of the cell cycle all the various hepatocyte nuclei are actually in. Thank you so much for listening, and I'd be happy to answer any questions that you have.